Hi, this is Manmad. Welcome back to MKF. Now we will discuss about aerobic plate count or total plate count in USFDA BAM method, Chapter 3, ISO method 4833-1. Just a comparative study. Both the methods, the media is same, plate count agar, but various time and temperature may vary. Why? See, in a BAM method as well as in ISO method, both are pour plate method. Means first we have to pour the inoculum after doing some serial dilutions, 10 to the power of 3 or 10 to the power of 2 or 10 to the power of 4. The colony range in BAM method is 25 colonies to 250 colonies is desirable to take as a count. Whereas in ISO method, 30 colonies to 300 colonies. That is okay almost. And media is same. Then why the temperature and time vary? Yes, let us have a look. In USFDA BAM method, so after inoculating the inoculum and sterile petri plates, then we have to pour the plate count agar media, sterilized plate count agar media, 15 ml or something like that. Have to keep, once solidify, you have to keep inverted position and keep it in the incubator. Means the temperature is 35 plus or minus 1 degree centigrade. The same process, whereas in ISO method, we have to keep at 30 degree centigrade plus or minus 1 degree centigrade. The time duration is, in USFDA BAM method, 48 hours plus or minus 2 hours. Whereas in ISO method, 72 hours plus or minus 3 hours. Why? In ISO method, incubation time is 3 days. Whereas in USFDA BAM method, incubation time is 2 days. And temperature also here 35 degrees centigrade. Whereas in ISO method, temperature is 30 degrees centigrade. Why? Why? Because at 35 degrees centigrade, most of the mesophiles, most of the pathogens are present. Most of the pathogens are mesophiles. At 35 degrees centigrade, we could able to know the mesophilic load, aerobic mesophilic load from the sample. Whereas at 30 degrees centigrade, means nothing but the room temperature, we could also able to know the count of not only aerobic mesophiles, in addition to the aerobic mesophiles, some spoilage bacteria and some psychrotrophs also we could able to know. Psychrotrophs, is it the word same as psychrophiles? We heard about psychrophiles. From 0 to 20 degrees centigrade, optimum temperature is 12 to 15 degrees centigrade. But what is psychrotrophs? Yes. See, psychrophiles, among the psychrophiles group, so some of the bacteria exhibits psychotrophic. Psychotrophs means they have the flexibility to exhibit the growth, and proliferation also will occur from the temperature plus 5 degrees centigrade to room temperature also. Up to 30 degrees centigrade also growth will exhibit. So that's why we called it as a psychotrophs. See, among 30 to 40% of psychrophiles contains psychotrophs. Examples of the psychotrophs, the best example is Listeria species. See, in Listeria isolation and identification process, at what temperature we will incubate? Primary enrichment at 30 degrees centigrade. Why? Because Listeria comes under psychotrophic. So psychotrophs which can grow at a temperature, room temperature 30 degrees centigrade. So that is why we have to incubate at 30 degrees centigrade plus or minus 1 degree centigrade. 3 days. Why 3 days? 2 days is enough, no? No. Because of psychotrophs will grow slowly. After 2 days only, the growth will exhibit. If you take the counting, if you take the plates for counting, okay, you may lose some amount of load, bacterial load. That's why you have to wait up to 3 days and you have to count. Not only aerobic mesophiles, in addition to aerobic mesophiles, we could also able to assess the load of the spoilage bacteria or psychotrophs. For example, if the sample source is same, Same sample you make it into two parts. One you did for USFDA BAM aerobic mesophiles analysis, and another one also you did aerobic plate count. Okay, BAM also you did aerobic plate count, and ISO method also you did aerobic plate count. If the same sample source in BAM, if you get cooked shrimps for example, the same sample source, if in BAM you will get 8000 CFE per gram, whereas in ISO method you may get minimum 10,000 to 12,000 CFE per gram. Means the TPC, the aerobic plate count load will be minimum 20% higher than USFDA BAM. If USFDA BAM aerobic plate count 8000 CFE per gram, whereas in ISO method aerobic plate count minimum 10,000 CFE per gram, means 20% growth, extra growth will occur. Why? Because minimum 20% depends upon the load of the psychotropes or spoilage bacteria in the product samples. Mostly shrimps are in seafood industry, so the chances of spoilage may occur because at psychotropes, due to the psychotropes only the spoilage may occur, because which can grow and multiply at room temperature or plus 10 temperature also there. These are the spoilage bacteria. Yersinica spoilage bacteria, Pseudomonas species also spoilage bacteria which can grow at room temperature well, and Listeria species also which can grow at room temperature well, etc. That's all about the video. Thank you, universe.